Okay, so this is review for test 1 for fall 2012. We're going to cover 4 chapters, chapter 1, chapter 3, chapter 4, and a good part of chapter 5. Okay, before we begin, I have to reiterate this point. This does not take the place of coming to class, and it does not take the place of your class notes. This is just to highlight some major points. Okay? So, we started with talking about the very basic core of life. That's atoms. Atoms are the basic building blocks that we use to make everything. Okay? We said that atoms have 2 roles, a structural role and a functional role. More on that in a second. That sucks. Okay. So, this is a test review for test 1. Fall 2012. Before we begin, I need to stress that this does not take the place of coming to class and looking at your class notes. This is just to highlight some major points, to reiterate some points, help you out some. But please do not think you can watch this video, come to the test, and be done with it. You have to review your class notes. Alright, so let's begin. We started this thing as a very basic level, and that's atoms. Okay, so we gotta know what atoms are, right? These are the building blocks of all substances. In class, we talked about the size of the protons and neutrons in the center versus the electrons around it, and we noted that that the electrons are much smaller than the protons and neutrons in the center. Okay, and that if we looked at the size, that most of an atom is just empty space. Now, we said that atoms have 2 roles, a structural role and a functional role. The structural role here is that atoms make up larger structures. For example, glucose is made of atoms. Our DNA is made of atoms. We are essentially made of atoms, and that idea is the structural role of atoms. It's like bricks to a building. We also said that there's these atoms involved in forming life structures. And you should know what these atoms are, and if you had a list like this on the test, you should know what does not belong there. So for example, if zinc was on there, and I might say, what atom is not integral in life, you would know that zinc, for example, is not. These atoms on bullet point 2, these are the major atoms we are made of. Primarily, we are carbon, nitrogen, oxygen, and hydrogen. Phosphorus we're going to see in DNA and our bilayer membranes. Sulfur we're going to see in protein. But the biggies, the important ones, are the first 4. Okay. Now, continuing with this idea of a structural role, atoms combine to make molecules. So I take 2 atoms, combine them together, I have just got a molecule. Now the second bullet point, we talked about this extensively later on, but you need to know that the 4 types of macromolecules that we are made up of. And all of these are just collections of specific arrangements of molecules. And we're going to talk about this a lot more later on. And here, these are examples of the macromolecules. Starting at the upper left, that is DNA. Upper right is protein. Lower right are lipids. Lower left are carbohydrates. Okay, atoms have another function other than being part of structures, and that is a functional role. What do I mean by this? The absence or presence of atoms is important to life. For example, you're an athlete. Or even just you're not an athlete, when your muscles contract. You have to have calcium present to have muscles to contract. Okay, so the presence of calcium, an atom, is important in the functioning of your cell to make your muscles contract. Sodium and potassium, when we look at the neuron, another good example of the absence or presence of a particular atom is important in life processes. Now, let's not worry about these examples on the bottom because we're going to talk a lot about this in the continuing weeks. but just lets know right now that the presence or the absence of an atom in the cell has a physiological effect on the cell. Okay, and these are just 3 examples of that. So what's the next level? Well, we're going to take those macromolecules, your nucleic acids, proteins, carbohydrates, and we're going to make up organelles. Okay. We're now reaching the cellular level. Now, organelles are just a collection of macromolecules. That's all they are. If I took all those macromolecules from before and combined them together, they are just a collection of macromolecules. Third point's an example of that. Do you see the orange structure on the lower left? That is a combination of the macromolecules we've discussed so far. The outer layer is lipids. Those little blue dots, those are going to be proteins. Not seen in this picture, but it's there, are nucleic acids. So organelles are simply just collections of the macromolecules we discussed before. Now, organelles combine to make up a cell. Next point is the cell is the smallest unit of life. Okay, I'll say that again. The cell is the smallest unit of life. Okay. Now, I want to make sure we see what we're doing here. We're going up and up a hierarchy. We started with atoms, we continued with molecules, then we went to organelles and the cellular level, and now we're going up to the tissue. So each slide as we progress through this is a higher and higher hierarchy of life. Okay? So, what's a cell? A cell is a collection of multiple organelles, at least in the case of eukaryotic organisms. Okay? And what happens is cells get together and form a tissue. Okay, so multiple cells interconnected, communicating with each other, they form tissue. Now, this point of intercommunication is very important. Okay, tissue they have their own genome, they're doing their own thing, but they have to communicate with other cells in the tissue to work. The analogy we used in class was what? Say you're in a band. Okay, you have your sheet music in front of you. Okay, well, let's say that let's make the comparison of a cell and its genetic material. That one player in a band is like one cell. The genetic material inside that cell is like the music that they're reading off of. But, if you've ever been in a band, you know that you have to hear all the other band members playing in order to be in unison with them. Well, the same thing here. This one cell has to get signals from the surrounding cells in order to be in unison and to become a tissue. Okay, so that's where that third point comes in. Tissue needs intercellular communication to work. That cell has its own set of instructions, but it's also getting signals from the cells around it in order to become a tissue. Okay, that's a very important point in biology and in tissue. Know what a tissue is. It's just several cells together to perform a specific task. Example is muscle tissue, epithelial tissue, nervous tissue, that's your brain, connective tissue. For the test, know the 4 types of tissue. Okay, and you can remember this as CMEN or C-MEN. Connective tissue, muscular tissue, epithelial tissue, and nervous tissue. Know the 4 types of tissue. Okay, so what do we have? We have got tissue, okay, and what if we combined 2 different types of tissue? For example, we can combine this connective muscular tissue. What would we get? We would get an organ. Okay. Now, an organ is just a collection of different tissues joined together to form a particular function. For example, here's the heart. What's it made? The heart's what? It's an organ. But let's say we pulled that heart apart, right? What would we see? Well, we'd see muscle, which is part of muscle tissue. We'd see connective tissue inside it. We'd also see nervous tissue, for example, projective fibers and this bundle of his in the center of it, which sends signals through it. So what's the point here? The heart is an organ, but what do we see inside of it? We see tissue. We see muscle tissue. We see connective tissue. We see nervous tissue. The point is this, that organs are collections of different specialized tissue. Okay? Any organ you look at, you're going to see multiple types of tissue. What are those types of tissue? Goes back to this slide right here. Those 4 types of tissue. Any organ, you'll be able to pull that organ apart and you're going to see these types of tissue. I take your lungs, pull your lungs apart. Your lungs are an organ. Guess what? You're going to see different types of tissue in your lungs. You're going to see epithelial tissue. You're going to see connective tissue. Okay? All organs are made up of differing types of tissue. What are those types of tissue? Connective, muscular, epithelial, and nervous tissue. Okay. So now we're at organs. We got one organ, the heart, the lungs, the stomach. Those are types of organs. The next level is organ systems. So these are when 2 or more organs work in unison, work together to do a particular function. Good example, the digestive system, right? It's your mouth, your throat, your stomach, small intestine, large intestine, pancreas. All of those are separate organs, but they work in unison. They work together. They work as a team to make an organ system. Okay, so what happens? We've got multiple organ systems. Some examples: reproductive system, your digestive system, okay, your lymphatic system. All of these are systems. These are 2 or more organs working together to do a particular function. The next level is an organism. This is all life, or excuse me, this is all of your organ systems combined to do a function. Okay, so for example, if I took your reproductive system, your digestive system, your respiratory system, we put all these together, we would make an individual person, and that's an organism. Okay, so let me just break here and kind of review what we did. And you have to know the order for the test. Okay, so just the point here is I want to show the order. I'm going to go through this kind of fast. We start at atoms. 2 or more atoms combine to make molecules. Molecules combine to make these macromolecules: nucleic acids, carbohydrates, proteins, and lipids. These macromolecules combine to make organelles. Second bullet point here. Okay? These organelles combine to make a cell. Multiple cells come together, communicate, talk to each other to form a tissue. We recognize 4. Multiple tissue types combine to make an organ. Multiple organs combine to make an organ system. Multiple organ systems combine to make an organism. You have to know the order present. On the test, you can probably expect to see a question where I give these different levels, and you have to know what is the correct order. Okay. Past the individual, we have a population, community, ecosystem, and biosphere. And again, that same idea is building upon each other. Many individuals are a population. Multiple populations are a community. That community and all of its environment is an ecosystem, and all the ecosystems combined is a biosphere. Okay, so that builds one on top of each other. So this part gives that thing we've just talked about kind of in a a nice, pretty little order here, and this is in the notes. Okay, and I'm just going through some things here that you can read on your own, obviously. Okay, so let's talk about homeostasis. Homeostasis is the idea that your body likes one condition and will try to maintain that condition. And in class, I've mentioned the idea of Goldilocks. And we said that what? What is Goldilocks? She likes something that's just right. The bed's too soft, the bed's too hard, but she likes it just right. The porridge. One's too hot, one's too cold, but one is just right. Well, our bodies, guys, are the same way. We like conditions to be one level, and our bodies will try to maintain that level. Temperature, great example. Our bodies like to be about 97, 98 Fahrenheit in there. 99, it fluctuates, but in there, about the high 90s Fahrenheit. So, what happens when we go to like 94 Fahrenheit? Do our bodies like that? No, they do not. What do? They're going to take steps in order to raise our body temperature. Good example. You come out of the shower, right? What happens when you come out of the shower and it's cold? What do? Well, what happens is you come out of the shower and it's cold and your body temperature goes down. You're away from that homeostatic level your body wants to be. So your body's going to take steps in order to raise your temperature. What's it going to do? One thing it's going to do is going to, what? Make you shiver. And that's going to warm you up. That's going to burn, it's going to make your muscles work, and it's going to warm you up. So that's why you shiver. Your skin is going to become clammy and cold. Why? Because your body is not shunting blood close to your surface of your skin. It's holding it back to maintain your internal body temperature more. Okay, the skin's like a radiator in a sense. And it's keeping the blood from the surface to prevent heat loss. That's why your skin becomes cold and clammy. Other side, what if you're out running real fast and you're really hot? What do? Well, your body temperature is going a little bit higher than it should. Your body doesn't like that. You're out of that homeostatic level. So your body's going to do steps to bring you down. What does it do? Well, first of all, it's going to make you sweat. That sweat is going to help take away heat energy. It's a heat sink. Your body's going to do another thing. It's going to do what? Your skin may become very red. Why is it becoming very red? Because the blood is going closer to the surface of your skin in order to shunt away heat from your skin, like a radiator. But the point is this, and here's the major point I'm trying to make. Your body likes a particular condition, just like Goldilocks liked a particular condition. And when your condition goes either too far, or too below that, or too above that, your body will take steps to get back to the condition it likes. And that idea of taking steps to return to the condition it likes is homeostasis. Right there, okay? It's homeostasis. Your body's taking steps to return it to that condition it likes. Body temperature is one we just talked about. The amount of carbon dioxide in your blood sugar, okay? It will try to maintain the condition it likes. Now, when you fall out of that zone, bad things happen, okay? Your body temperature goes too high, what happens? You have heat stroke and you die. Your body temperature goes too low, what do you get? Hypothermia. You die. Blood sugar, another good example. If it goes out of whack, like with someone with diabetes, you can't maintain that homeostatic level, what happens? Goes too far, you can go into a diabetic coma and die. So there's a good reason you want to maintain homeostasis, because the further you get from that happy zone, okay, the closer you are to death. Okay, and there's a reason why your body does that. Now, we talked about homeostasis, but how does your body know all this is happening? How does your body know the temperature is changing or your blood pressure is changing? Well, it just so happens that we have receptors in our systems that are going to detect these things. Okay, we've got receptors that detect the temperature, they detect blood pressure, baroreceptors. We have receptors that detect chemicals. So our bodies are monitoring. Actually, as a matter of fact, we have receptors in our joints that monitor motion, and the more motion is relayed to our brains, and that will lead to increasing in oxygen intake. Point's this. Here's the point, guys. We have receptors in our body that detect these changes. Temperature, blood pressure, chemicals, many different changes. And when it detects a change, what does it do? We just said it. It takes steps to bring it back to the correct level. Let's look at those thermoreceptors. Our body detects that it's getting too hot. What does it do? You sweat like a pig, right? You put your blood closer to the skin and it becomes very flush, but those are steps to bring your blood or your temperature down. Thermoreceptors detect that you're too cold. What does your body do? Those receptors communicate the information to the brain, and then your brain says, okay, let's correct that. We're going to make you shiver. We're going to pull the blood closer in, so it's going to make your skin clammy. Total effect, raises your body temperature. Okay, that's the point. Receptors are necessary for homeostasis because they detect the change. Your body wants to maintain a level, but how does your body know what level is what? That's the role of the receptors. The receptors detect what your body levels are, and they are going to assist your brain to return you to your correct level. Okay. Okay, and you can read this for your own. You can read this for your own, obviously. Finally, I want to talk about scientific naming. Biologists love naming things. You know, we love naming things. And we have a way, we call it the binomial naming or nomenclature system. Okay. Genus and species. First of all, know what a genus is, know what a species is. In this naming system, we have genus is first, species is second. A good way to remember this is genus is more general. See the gen and general? So genus is more general. Species is more specific. Good mnemonic to remember this. General, specific. So for example, we're Homo sapien, right? Well, Homo is a more general term. You can have Homo erectus, Homo Neanderthal, Homo sapien. Sapien is more specific. It narrows it down to one particular species in that genera, or that genus. So how do you name scientific names? Well, first of all, you have to italicize the name, okay? Or you can underline it in some cases, but it's italicized. 2, the genus is capitalized. And 3, the species is lowercase. Now, many times in scientific literature, once you name a full species, or a genus and species, you can abbreviate the genus with just the first letter. For example, E. coli, or you might say B. anthracis for Bacillus anthracis, or H. sapien for Homo sapien. But you should know how to do scientific naming. And you can read this for yourself. We won't worry about that, but we'll talk about that a little bit later. Okay. And I'm just skipping past these because I want to make sure I can test what I'm skipping past, but I just want to hit the high points. So you can read this for yourself, and you can read this for yourself. Okay. One thing I want to talk about is the scientific process. You should know the order of the scientific process. This is the scientific process. This is how, let me say it this way. We don't make this stuff up, okay? When we do science, we have a order of operations, and this is the scientific process. You should know the order of the scientific process and a definition for each thing, okay? On the test, I could even put this in a order and say, what's the correct order? And I might say observation, question, hypothesis, prediction. And then, assess results and report. But then I might say, is it question, hypothesis, observation, prediction? No, that's out of order. You should know for the test the correct order that it comes in. So the first is observation. This is when you see something, okay, and it's like, I don't understand that. Like a what the heck moment's going on here. Next is a question. It's more developed than the observation. Why does this do that? Type of things. Okay. Third, you need to make a testable hypothesis. Okay, very important here. You have to have a testable hypothesis. If it's not testable, well, is it really science? That's a question. If I can't test something, then it pretty much ends here. Okay, I could say I think the planet Neptune is made of blue cheese. Well, can I really test that? Not really. You know? So you really need a testable hypothesis for science. Okay. You can use computer models, you can use actual empirical data, it depends, but it has to have some way to test it. Next is the prediction. Now we make these in the form of if-then statements. Okay? If X, then Y. That's a prediction. Okay, so we've made the prediction based on the hypothesis, and then we do an experiment. And you test that hypothesis. Key point here is it has to be repeatable. An experiment has to be repeatable because when you report it, step 7 on the list on your right, other scientists will want to do the same thing. And it has to be repeatable because if I do an experiment in my lab, someone from another state or another country might want to do the same thing. And if they don't get the same results, well, that's a problem. Okay, so it has to be repeatable by yourself and by other scientists. And again, objectivity here is incredibly important. Okay, because, as scientists, we try to be objective, but we're human beings. At our core, we are just humans. And we want our hypothesis to be correct. We really, in our heart of hearts, want it to be correct. So when we do experiments, we really, to ourselves, stress this idea of objectivity. Okay, we try to separate ourselves from the information and try to make sure that we analyze the data in a non-biased way. Some people go as far as to collect the data and have someone else run the information just to make sure our objectivity does not come into it. Okay. Next is assess results. Kind of what I said before, we take the data from the experiment and we do an analysis. Some people go to this extreme of having another person, assign unknown numbers to a set of data. For example, I might have collected data from 2 different conditions, right? And I would take it to another person and say, here, code this data so I don't know what data came from what. And then they would give it back to me and say, okay, A is this, B is that. And then by that way, I don't know what is what, and then I'm not tempted to put my own subjectivity into the data. Okay, so we got to assess the results. Then we got to report. Okay, we got to take what we have and tell the world. Because if we don't tell anyone, who's going to know? Well, the answer to that's no one. So we got to report the data. Okay. An experiment, you should know, is a test that can support or falsify a prediction. A variable is a single characteristic that differs between the control group and the experimental group. Before I talk about that, let me just go to the next slide because we talk about control groups and experimental groups. And I'm going to bring that point right back one more time. So an experimental group is what you're testing. Well, let me say it this way. Let me step back here. Let me give you an example to make this point. I'm part of a pharmaceutical company, and I have a new blood pressure medication. Okay? I want to test this blood pressure medication to make sure it works. in the lab in controlled conditions it works, but I don't know if that works in human beings. Okay, so the test that I'm going to do is going to be, if I give this test, or if I give this drug, will it reduce blood pressure? Okay, so that's my experiments. I'm going to give this to different people to see what result it has. Now the variable is, if I give this drug, let me say the variable is the presence or absence of the drug. Okay, so I could say if I give this drug to people, their blood pressure will be reduced. Now, if I give this drug to 100 people, how do I know that that drug is effective? I have to have a single variable. I have to have 50 people that I give this drug to, and then I have to have 50 people that I give a placebo, a sugar pill to. And I want to see if I change just one variable, the presence or absence of this drug, will that make a result? So let's just set this up. I would have 50 people I would give my blood pressure drug to. Another 50 people I would give a placebo or a sugar pill to. And I would watch what happened to their blood pressure over the next 6 months. At the end of that 6 months, I would say, okay, experimental group, it went from this level, and then it reduced by 10%. The control group, it went from one level, and it reduced by 1%. Well, that would tell me that, most likely that experimental group blood pressure drop was because of the presence of my drug that I'm testing. Okay, and this just keeps on talking about what we're talking about. You can read that yourself. And that concludes Chapter 1.